Hello, I'm Mary Gehring, and I'm an assistant professor here at MIT in the biology department, and I'm also a member of the Whitehead Institute. So I came to MIT about 2.5 years ago in 2010 after doing a postdoc in Seattle. And really for my entire career I've been interested in plant biology and plant development. This probably comes from growing up in a very rural part of the country in northern Michigan, where I was exposed to the outdoors a lot, and I was always very interested in the natural world. And so when I was in college as an undergraduate, I started doing research on a plant called Arabidopsis thaliana. This humble little plant, Arabidopsis thaliana, which is actually a weed. It's part of the mustard family, but it's a weed. You might find it growing on the side of the road, and it grows all around the world, in Asia, in the Americas, and in Europe. This plant was the first plant that had its entire genome sequenced. So we know all the basis of its genome, all the A, T, C's, and G's. And that's very powerful information for understanding the biology of plants and of really all organisms. And so some of the characteristic of model organisms like this plant Arabidopsis and other organisms that you'll be hearing about during the class, like fruit flies and yeast and worms, are that they're very easy to work with. So as scientists, we come up with scientific reasons for using organisms, but also very practical reasons. So this plant Arabidopsis, for example, as you can see, it's pretty small. It fits very nicely in this small pot. It has a relatively quick life cycle. So as scientists, we always want to be doing experiments and getting our data as quickly as possible, and then analyzing our data and coming up with new hypotheses and testing those hypotheses. So we like to use organisms that have a fast life cycle. So this plant, for example, completes its life cycle in about 6 to 8 weeks. So that's really great for us. So model organisms have been chosen by the scientific community to focus our attention on, and from studying these relatively simple organisms, derive principles about the larger diversity of life. Some of the other important features are that we can make mutations in this plant. So we can induce mutations. So we can change the sequence of the genome, the A, T, C's, and G's, and we can come up with mutants that have different traits. And from that, we can learn what genes control those traits. So you've heard in the lecture today, or recently, about Mendel's peas and how Mendel used crosses between two different pea plants that had different characteristics in order to really discover the nature of inheritance. And these crosses between plants that had, for example, purple flowers and white flowers allowed him to discover that there were things called genes. And from this work, he was able to develop the laws of inheritance. So it was a number of years where Mendel's research was really lost to science. And then in the early 1900s, people discovered Mendel's laws, and using organisms like corn and fruit flies were able to really determine the nature of genes and the nature of chromosomes. In addition to growing these plants in a pot, you can also grow them on agar plates like you would bacteria. So these are young plants that are a couple weeks old growing on these plates. And because you can do things like this, it makes it very, very easy to work with this organism and to grow up lots of plants and to look for phenotypes like Mendel did that you might be interested in understanding what are the genes that underlies those phenotypes. So whereas Mendel looked at traits of flower color and pea size and whether they were wrinkled or smooth, we can take these Arabidopsis and grow up large quantities of them and look for whatever phenotype we might be interested in, like root length or color of the leaves. And so for these reasons, Arabidopsis is a great model organism. So it's the plant model organism, and it's really a very powerful tool for genetics. So even though Mendel started out with peas to understand what genes are, now we're still using plants to understand the nature of genes and the nature of inheritance.